Health Center Munich in Germany, and his talk is about the crop improvement and pangenomics. Dr. Spanigal, please.
Thank you very much. I hope you can see my screen now.
Yes, we can.
Looks good? Okay, perfect. Let me get that going. Just a second. Okay. Right. I also want to thank Molecular Plant for organizing that event. I think that is really fantastic. And I also want to thank the committee for giving me the chance to present my work today. I hope you're still fresh after the talks. And we're going to switch a little bit gears now, because I'm going to talk about plant pangenomics and how they can actually help to improve our most important or our emerging crops. And first, I want to take you to a little time journey back in the history of plant genome sequencing and start with some reference genome stuff. If you think reference genomes are a little bit old-fashioned, don't worry. I have something for you as well in the second part of my talk. And here's a timeline about the history of plant genome sequencing. So with the start in the year 2000, with the first ever sequenced plant genome, Aropsis thaliana, there was some quite nice progress over the last 20 years. So we've seen the genomes from rice, maize, tomato, and more recently, also the genome of bread wheat in the year 2018. So looking at that, you may wonder, well, why did it take almost 20 years to get the genome of bread wheat decoded? And the reason for that is actually the size and the complexity of the wheat genome. Bread wheat is an allohexaploid. That means it consists of 3 different subgenomes, here called the A, B, and D subgenomes. And these subgenomes are full of repetitive elements and transposons, and they are highly similar between each other. And I always like that schematic representation of the wheat genome to illustrate the dimension that we are talking about here. For example, if you're going to compare the human genome to the bread wheat genome, it would only make up these 2.5 chromosomes over here. Aropsis thaliana, only the tiny little piece. The rice genome, half a chromosome, and also the genome of maize would make up 2 chromosomes here. So nevertheless, 2018, I told you, the International Wheat Genome Sequencing Consortium actually succeeded in reporting a fully annotated and ordered genome sequence of bread wheat. And that was a starting point. It was really a game changer for research in bread wheat. So I will talk a little bit more about wheat in the second part of my talk, but now I want to switch to another crop, which is really a special crop, and I will tell you why. It's oat. Oat is a little bit of an underutilized crop so far, but it has some very substantial and unique benefits. First, it can help to prevent cardiovascular diseases. It's gluten-free. It is high in beta-glucans and fibers, which help to stabilize your glycemic index after meals, for example. It's a very nice protein source and can replace meat-based products. It requires fewer fertilizers and pesticides compared to other crops, and it has a low carbon footprint. So all of that makes oat a really emerging and special crop. And you may wonder, well, where is the reference genome sequence for that? And here it is. So what we did here is we generated a reference genome sequence for the hexaploid oat. Again, we are talking about an allohexaploid with 3 different subgenomes here, very similar to the situation in wheat, here called A, C, and D. And the ACD is the hexaploid. And in a collaboration headed by the Scan Oats Consortium in Sweden and headed by Olof Olsen and Nick Siljowski, we sequenced the cultivar, the Swedish cultivar Sang, and in collaboration with partners from the US and Canada, We also generated reference sequences for the tetraploid, Avena insularis over here, and Avena longiclumis, the diploid progenitors here. So talking about the genome assembly and the annotation, the genome assembly in total summed up to around 11 gigabases. You can see also a quite large genome here. We predicted over 80,000 high confidence genes and evaluated that using conserved ortholog measures called BUSCO. The higher the BUSCO, the better usually. And BUSCOs for the assembly and the annotation over 98% indicate high completeness and accuracy of the genome sequence. So we got that published last year and had the chance to have this really nice oat plant on the cover of Nature, which I think makes a very nice cover. And I want to take the chance now to give you a few insights into the fascinating oat genome. So what you see here is a schematic representation, and all the lines indicate syntenic relationships between the subgenomes of oat. The ones in more bold color, these represents the syntenic relationships between chromosomes not from the same linkage group. So for example, syntenic relations between the chromosome 7D here with the chromosome 2A. If you compare, so you already see that a lot of things is going on in terms of rearrangements between the subgenomes. If you now compare the same picture with that in bred wheat, you can immediately spot the difference, I guess. Whereas in oat, you can see kind of a lot of things going on between the subgenomes. In wheat, there is actually very little going on, almost nothing. And this is also supported by this visualization that was done using Synvisio. So you see, this is the oat genome, the ACD subgenomes here. And you can see there's really a lot of rearrangements within the subgenomes, but also between the subgenomes. So we were making up our mind, why does oat have so many rearrangements, especially compared to bred wheat? And in bred wheat, there is a factor that is involved, especially in that, and it's called PH1. The PH1 locus promotes homologous crossover and prevents homologous crossover. So this is kind of a tricky wording here. That's why we have this illustration down here. So when you talk about the two chromosomes here from the same subgenomes, that's called the homologs. And when you talk about the chromosomes, for example, chromosome 2 here from the A, B, and ABC subgenomes, in that schematic representation here, you're talking about homologous chromosomes. And this PH1 locus is actually researched since over 15 years, and it's a duplication from chromosome 3B to chromosome 5B in wheat. And recently, that has been narrowed down to one single gene called CYP4. So now we were actually wondering, well, does oat have this CYP4 ortholog? Is there also a PH1 locus present in the oat genome? And that's a synteny plot here. And what you can see here, these are the syntenic regions between oat and wheat. And what you can spot is there is a very nice syntenic relation upstream and downstream of the PH1 locus of CYP4, but exactly not in the CYP4 locus, and oat has no ortholog to wheat's CYP4. So what we find is the CYP4 duplication is really unique to wheat, and oat is an example of a polyploid genome without the CYP4, which we think contributes to the observation that I showed you before, that oat has so many more rearrangements. It's certainly not the only factor. Also, divergent time and other factors will certainly play a role in that observation. But the CYP4 is certainly also an important factor. So this new reference for oat really opens the door for new opportunities in oat research and especially breeding. So the identification of breeding barriers is now possible after characterizing all these translocations. So they have so far complicated oat breeding a lot. and now it's much easier by knowing about them. So what we also did in that study is we cataloged, deciphered, and analyzed gene families that are related to these specific traits and properties that I showed you about oat, for example, the nutritious qualities of oat. And another thing is that we are now able to identify genes underlying agricultural relevant traits for genetic breeding, and that's the title of my topic. So I want to show you an example of how we can do that. The collaboration partners from ScanOats, Nick Syrovsky and his team found in a tilling population a mutant phenotype that has a glossy inflorescence. So this mutant line actually has no epicutical rex, and that you can see here in that very nicely in that micrograph picture here. So these vexes are very important in biotic and abiotic stress resistance and are therefore a prime target for oat breeding. So now that we had the reference sequence in hand, of course, we were wondering, can we actually find a locus or a mutation or a candidate gene that is causative for this mutant phenotype? And that's what the colleagues did here. So we sequenced a voxy and a glossy genotype, and also pools of homozygous glosses and homozygous glossy plants. We actually also found another mutant line that showed the same phenotype there, and looked for candidate genes that actually showed a difference between the wild type and the mutants. And what we found is a candidate gene that is orthologous to the barley SIRQ gene, and this gene is involved in the beta diketone VOX synthesis. So this was our prime candidate actually for this phenotype that we observe here. And by looking at this alignment, you really see the mutations that are making up the difference between the glossy and the waxy phenotype. So now, if you remember, we are talking about an allohexaploid here. That means in theory, you have always like 3 copies of a gene present in your genome, right? And the same is also true for the voxy gene here. So you're going to have homologous clusters on chromosome 2C and 3A, in addition to the SIRQ candidate that we identified on 1C. But when we looked at the expression of these other genes, we found that basically only that one on 1C was expressed and the others were not expressed or not functional. So this is really an example for a functionally single copy gene in oat. And these are also prime candidates in general for breeding applications in wheat and also oat. So the oat genome is now available in Ensemble Plants for those of you who are interested in having a look and browsing around a little bit. So it's available there. And there are all other oat genome resources out now since last year. So there is a Chinese variety, Sanfenzan, I don't know if I pronounced it correctly, published by Peng et al. In Nature Genetics also last year. And there's a long read assembly by PepsiCo and Corteva from the US available. And these resources are all available in grain genes. So now, I've already talked about additional resources, so that already looks like a mini pan genome, right? And that's the logical next step that we took now that we had a reference sequence for oat. We initiated a pan genome project, and that's the Pan Oat Consortium, headed by Martin Mascher and Nick Tinker. And you can see that there's quite a number of people coming together, indicating some good interest in oats, which is very nice. And the Pan Oat panel actually is set up to capture most of the global oat diversity worldwide. That's one of the main objectives that we have here. And the other objective is that we also want, of course, capture genetic diversity. What you see here is a PCA of the global genetic diversity of oats. And you can see that we picked varieties that represent the different main clusters. But also zooming into the spring oat block here, we've made sure that we really capture the different varieties and have representatives of these included in the Pan Oat panel. So one thing that I want to stress here is why is that all possible now? And this is really because of the new technologies that made a dramatic leap forward within the last 5 years, and especially also in the last 2 years, based on the accurate long reads that are provided by hi-fi sequencing from PacBio, which allow us to tackle even the most complex plant and crop genomes now and get assemblies with up to 50 megabases contigs. And the second revolution that we see now to be able to assemble these into full chromosomes is the high-C chromosomal linkage data technology that helps us to scaffold all these long reads and long contigs together. So this is really a game changer now for doing genomics, especially of the complex and large genomes out and read. So what we delivered now in PanOut is 28 fully annotated genome assemblies. You can see a total number of around 11 gigabases in size, and the mean N50 is quite impressive, with 88 megabases here. In average, we annotate more than 100,000 genes on each of the cultivars that are sequenced now. And now we are in the process of doing full comparative analysis on top of that, on that set. So now I've already talked and used the word pan-genomes multiple times. So now I think it's time for a more formal definition. What is actually a pan-genome and what are pan-genomes good for? How can we use them? So pan-genome, well, one single reference genome is a big help, is a big step for each community, but it's not enough. It just represents a single view on a genomic landscape. So additional genomes that come into the play and that are compared with the other existing genomes make up a pan-genome. So there's no certain number, but usually you're going to have a couple of genomes that are coming together to form a pan-genome. And when you then look at the gene inventories of these and compare them and look at the gene inventories, you're going to have a core gene set here, which is shared by all the genomes that you have here. You're going to have a cloud, which is exclusive to one or more of the genomes. And then you have a shell, which is shared by only a few or some of the genomes here. And all of these have different properties. So the core genome is usually enriched for essential cellular processes, whereas the cloud and shell are more usually enriched for processes that may be only beneficial under only some environmental conditions. For example, you would see stress resistance genes in some cases amongst these, and these are of course then very interesting for crop improvement. So in general, saying pan-genomes really help to access the natural genetic diversity of plant species and help to improve crops. And I want to give you an example now coming back to wheat, where such a pan-genome actually delivered exactly that promise. So the 10+ Wheat Genome Project, headed by Curtis Posniak from the Crop Development Center in Saskatchewan. In that consortium, we sampled 10 different wheat varieties, harnessing really the diversity from global breeding programs, and that was published in 2020. The main criteria for selecting the cultivars in that panel were really breeder-driven. So we were looking for allelic diversity, founder lines, high priority genes, but also existing genomes. And examples for that are that some of these lines had a specific introgression that is also associated very often with resistance traits, insect resistance, but also known large-scale translocations that are important for breeding. So as I told you, we had a special focus on harnessing diversity from global breeding programs, so no wild weeds or so were included, but we were really looking to represent the breeding programs worldwide in that panel, what you can see here. And I just want to give you one example. where we looked into large-scale structural variation. I already told you large-scale structural variation is very important for breeding purposes. And this is a gene space plot amongst all the different 10 read lines separated into their subgenomes. And if you look close enough to that one, and I will highlight that to you, you see here in ARENA LR4, a Swiss line, you see like there's a reshuffling in the lines down there. And this is actually a translocation. Whereas the wild type chromosomes here are 7b and 5b with no breakpoints. These translocated chromosomes have a certain breakpoint here, and the translocation between 7b and 5b is present in a few cultivars. So we didn't want to stop. The next step after a pan-genome can, for example, be a pan-transcriptome. That's what we are currently doing together with the Earlham Institute, headed by Anthony Hall, and us, and the team in John Innes generated all the data here. So we had isoseq data from 14 cultivars, 5 developmental stages and seedlings from dusk and dawn, making up more than 250 samples here. And these were grown in the same conditions and now compared. And they were used to deliver full de novo gene predictions, which are then a step towards a functional pan-genome, looking at variation expression, alternative splice sites, and so on. This is again available in Ensemble Plants in case you're interested for download and browsing. And I will show you one example about the orthologous gene groups. So these orthologous gene groups are really set up compared between the different varieties. And if you look close enough, most of the gene groups are actually shared between all the 10 lines. But then there are some examples where you're going to have clusters with only two varieties present. Here, for example, you have a Canadian cluster, orthologous groups only from with lines from Canada, and the same for Australia, and so on and so forth. So these groups are prime candidates for interesting function of adaptation. One more thing that I want to show you here is looking at triads. Triads are the orthologous genes between the A, B, and D subgenomes. So you can think about the copy of the same gene across the 3 different subgenomes. And looking at them, you can either define them as stable, and that means they show the same expression across all the 10 cultivars, or they are dynamic, and that shows they have a dissimilar expression across the 10 cultivars. When you look at the stable triads, most of them are actually here in the balanced gene count, and a balanced gene count means, I don't have time for a formal definition now, but a balanced gene counts basically mean that you have similar expression in the A, B, and D copy, okay? For example, if you have a B dominant one, then the B copy is much more expressed than the others. So most of them are actually in the balance, but you also have a consistent bias in all the different varieties, for example, of D dominant triads here. When you look at the dynamic triads, you see, well, there is much more going on. So they are not always here in the more balanced area, but they are really spread towards the end here. What you also found, and I can show you here, is that these stable triads are not evenly dispersed across the chromosomes, so there are hotspots for that, and we are looking into the causes for that. So one more thing that I want to show you here is one very nice project that we are currently running together with colleagues from the John Innes and also the Earlham Institute. And that goes back to the history and the evolution of the triticale. As I told you, bread wheat is an allohexaploid, and it originated from a hybridization of the tetraploid A and B, which you also know as the durum wheat, and the D genome Aegilops tauschei. The nice thing is that you can actually generate synthetic wheats by hybridizing durum wheat with the D's genome here, Aegilops tauschei. And that's what you did here in that project design, and you see that here. So that's the tetraploid genome donors. And we generated synthetic reads with two different independent D subgenome donors here. We generated triploid plants, A, B, and D, and then consequently, we generated fully functional A, B, D hexaploid read plants here. And why did we do that? And what are our main objectives, or what they are interested in? So we are interested, what are the contributions of chromatin changes to triad gene dosage and altered gene expression? And we also wondered, how consistent are these epigenetic changes in the independent synthetic lines? And do these genomic interaction actually influence gene expression? So that's the experimental setup and analytical space. So you can see we actually generated a lot of data, and I can only show you one result here from the gene expression analysis today. But we also generated chromatin, accessible chromatin data, gene methylation, and also histone modification data here. And that's the only thing I want to show you today here. When you now compare gene expression between the triploid, that's in the middle here, the diploid, and the tetraploid, you can see that most of the expressed genes in the D in the diploid are actually going to the balanced state here in the triploid. But there is also a number that are getting then suppressed in the triploid from the expressed genes in the donor. From the other side, most balanced A and B gene pairs remain balanced here, but also some of them go into the other categories here. So you see there is a lot of dynamic going on here, and now we are looking into the epigenetic marks in the course of that project. And with that, I want to finish up here by just acknowledging a few people, because these things are really huge team efforts, and I'm incredibly grateful for the support of the consortia and people here, especially Martin Mascher, Nils Stein, a long-standing collaboration, Nick Siriovsky for the OAT, and the 10-read genome project headed by Curtis Posniak, and then the ERA-CUPS project with Mike Bevan, who's a fantastic has been a fantastic mentor for years. Anthony Hoare, great collaboration partners, and of course my group, and especially my director Klaus Meyer, who supports me since ages. So with that, I want to thank you, and happy for any questions.
Thank you, Dr. Spanegal. And now it's time for the Q&A session. Any question from our committee members?
Yeah, I was quite fascinated by the PH1 locus, and I was not aware that it has been, mapped down to a single gene. And one thing I was wondering then is, because you kind of like went over this a bit too fast for me, but you said there is a duplication, and so does it mean that it's a gain of function, that by increasing this CYP4B2 dosage, you can actually prevent the homologous pairing? And would that be maybe transferable, for example, into your OAT genomes?
That's an excellent question, and I'm sorry for being fast on that. I noticed I was a bit fast on that. Yes, that's actually so this it's a transduplication. So you're going to have 3 copies of the originating genes in read, and then you have an extra copy which has been transduplicated to the chromosome 5. And this transduplication is not present in OAT. It's only there in read. And I think one of the theories is that you have kind of a dosage effect there by this additional copy, because it's expressed.
But this duplication is then in all the 3 subgenomes, or only in one?
No, only in one. Only in one. It's so you have the 3 copies, and these 3 copies are also there in OAT. But in read, you're going to have one additional copy, and that's the CYP4 referred to, and that's on chromosome 5. And of course, now it's a very fascinating idea to bring that copy also to a system like the OAT genome and see what happens. And actually, that's something what we would love to do, and see actually if it also prevents homologous pairing in a system like OAT.
Yeah, thank you.
Okay, okay, thank you, Menu, next talk. So I have a question. So many wheat cultivars have the integration from the wild relatives or from other cereal species. So when you construct and analysis the pangenome of the wheat, how do you differentiate the integration and structure variations?
Yeah, that's a very good question, because these introgressions are actually also prime targets for the selection of cultivars to be sequenced, because very often, as I said, they are carrying like genes of high interest, like resistance genes or other important traits. So there's different methods for identifying the introgressions actually in such a panel. One of them is actually what I just showed you in this orthologous plot, where you find suddenly groups of genes that are shared only, for example, by cultivars originated from Australia or for Canada. In many cases, we were able to show that actually behind these clusters of shared genes that are only present in a geographically distinct area, introgressions are the cause for that, because then they are also clustered on chromosomal regions. And then you can really trace them back to an introgression event. But there's also other methods out there to identify these introgressions, or to already in a panel of germplasm to select for lines that have these introgressions. This is a prime target for crop improvement for sure.
Okay, thank you.
Okay. Oh, yeah, sorry. Go ahead, please.
Can I just very general question is in the beginning, you nicely convinced us about the potential of oat as a crop with a lot of advantages, but there are also kind of problems, I think, because first of all, bread making with oat will be difficult without the like. But secondly, I think oat is also kind of northern hemispheric species. I don't know of some successful oat cultivation in northern Africa or whatever. I don't think so. Do you think that your pangenome approach might come in here, help here to, because if you consider climate warming, and might be that oat is going to get it more and more difficult in the coming years, probably.
Yes, you're absolutely right, and that's exactly a project currently. My postdoc and first author paper on postdoc on the paper is currently conducting. So we are trying to get oat more drought resistant, so that it can also be grown more widely, like in climates of the Mediterranean area, or you say like Northern Africa or so, because it has the potential to get a stable crop actually. So from the benefits and so on, it's a fantastic crop. But you're completely right. So there needs to be research to get oat adapted towards drier and warmer climate conditions. That's definitely something which is very tempting and needs to be done at the moment. And as I said, so there are proposals out now to do exactly that. And the genomic resources that we have, they are really the basis to do that now.
Okay, thank you.
Okay, any questions from the committee members? Okay, then thank you again, Dr. Spanigal.
Thanks.
And that also calls the end of day one of our RSPS Award 2023. And we want to thank again all the presenters for their nice talks, and all the committee members for their time and participation. And also thank you all to all the audience who has been watching our live stream from all different platforms. And we sincerely hope you have enjoyed the science. And before we leave, just a short reminder that we do have another oral presentation session tomorrow, and another 5 outstanding young scientists will be sharing with us their very interesting research stories. So please come back, and we will see you all tomorrow. Yep.
See you.
Bye.
Thank you, Wenjia. Also, thank you, Christine. Thank you, Tom. Yeah, thank you all for joining this meeting.
Yeah. Thanks a lot. See you.
See you tomorrow. Bye.